Hello, and thank you for joining me today. I'm Martin Latterich, the Chief Scientific Officer of Bioscale. Bioscale is a company offering integrated solutions in developing biomarker immunoassays for use in basic and clinical research. Before I delve into my presentation, I'd like to declare a financial conflict of interest as an executive and shareholder of Bioscale. Furthermore, I'll mention biomarkers associated with human disease and technology for biomarker discovery. By no means do I make claims as to their suitability and use as an approved diagnostic. With the legal disclaimer out of the way, I like to focus on what is rapidly becoming an evolving medical practice and a paradigm shift that goes from classical medical practice to the future of medicine. So in the so-called 20th century empirical medicine, you would go visit your doctor with an ailment or illness. The doctor would perform a symptom-based diagnostic, possibly an in vitro diagnostic or X-ray imaging, and then based on the physician's experience, the physician would prescribe a treatment. Very often, for example, when you go see a physician, the physician would say something like, oh, you have a sore throat, it could be bacterial, here, try this antibiotic. If it doesn't work, come back in a few weeks, and then we'll try something different. And very often you would either recover or revisit your physician. Where medical practice is evolving in the 21st century, so-called the precision medicine paradigm, you would visit your physician. The physician would already know your genetic profile and would diagnose phenotypic links to disease. The physician would basically say, well, we can monitor your progression of disease, or we can prescribe some preventative programs, and then you would have a targeted diagnostic assay or an in vitro diagnostic assay or imaging, which enables the physician to prescribe the relevant treatment. And then during the treatment, you would use again a in vitro diagnostic or imaging to monitor the progression of disease. All this is very, very relevant to cancer, because cancer is a genetic disease that basically proceeds with the development of a tumor which is due to a genetic aberration, and then the tumor will grow out of control, and all the tumor development and tumor growth is associated with changes in the molecular makeup of both the genome as well as the proteome of the tumor. And so I think where medicine is rapidly progressing is there's a much broader rational basis that is developing in order to precisely understand the cause of the disease and hopefully have the relevant therapeutic that goes along. So that is basically what is known as precision medicine. And so very important for the paradigm of precision medicine is the actual ability to diagnose a condition, and this ability is basically driven by biomarkers. Biomarkers are molecules that have an association with a specific disease. For example, cystic fibrosis is caused by a mutation in a chloride channel. And so the aberrant gene of the chloride channel is a biomarker for the disease like cystic fibrosis. Not all biomarkers are genomic in origin. Some of them can be proteins or small molecules. For example, a good biomarker for a hemoglobin disorder such as beta thalassemia is actually the protein, the hemoglobin itself. And so a biomarker can also be a protein that is either causing the disease or that is associated with the disease. You make a discrimination between biomarkers that are mechanistically linked to a disease. These biomarkers are generally mechanistic biomarkers. They're directly involved in the disease process, or biomarkers can also be associated with a disease but not causing the disease. And these biomarkers would be termed surrogate biomarkers. Now, how do you go on discovering biomarkers? So in very simplistic terms, you would look for differences between healthy individuals or diseased patients, and you would see what are the molecular differences between both patient and healthy individual. And so it's a very simplistic paradigm to go screening and looking for differences. Now, at the same time, every human being is molecularly very distinct from one another. They share very similar genomic makeups, but at the same time, they are exposed to different environments, they're exposed to a different history, and all of this can cause differences in the molecular makeups between patient and healthy individuals. And so not all differences between healthy individuals and patients are actually biomarkers for the disease of the patient. And so the generally accepted definition of biomarker discovery is finding differences among, for example, messenger RNA, proteins, protein modifications, and metabolites between health and disease is actually quite easy. So it's very easy because we have outstanding molecular tools that allow us to measure these various biomarkers. However, and that is something really important to remember when you go on and discover biomarkers, many of such differences are random or nutritional. Very few differences can be validated across many patients and are functional or surrogate biomarkers. And that is actually one of the reasons why in the last 20 years or so, actually there are not very many new biomarkers that made it into a clinical practice. And that was in part due to the either technologies not being available or the emphasis on validation wasn't really there. And so it's only in recent years that we see a new a number of biomarkers for disease that are being coming to the market today. So what is needed for precision medicine are biomarkers that can predict the disease or outcome or diagnose it at an early stage. Simply looking for differences is not enough, but the discovery actually has to be followed by extensive validation. And so when you look at the traditional biomarker research pipeline, you can discriminate between discovery phase, you can discriminate between validation phase, and then ultimately the desired outcome of a biomarker discovery and validation effort is actually the development of a diagnostic assay. And so there are different, fundamentally different tools that are being employed to discover biomarkers, that is, looking for meaningful differences between healthy and diseased individuals. Some of these solutions are, or most of these solutions are actually mass spectrometry based, because the mass spectrometer is an excellent platform to interrogate molecular differences between two different samples. And some of the last decade has focused on optimizing the technologies to statistically significant, to describe different biomarkers that are statistically significant. So looking for statistic differences between, say, a healthy tissue and a tumor tissue. Some of these methods, which I will mention later on, relate to iTrack, which is a method, say, a mass spectrometry method that employs isobaric mass tags that allow you to trace materials and peptides and proteins from different samples. But it's only one method out of many different ways to actually look at the differential proteomic makeup. And then there are somewhat other methods, for example, protein microarrays and cell profiling, which were quite frequently used in the past, but which are currently being displaced by more accurate solutions. The validation phase is divided into two sections. The first one is an analytical validation, which means you're actually confirming that what you measured as a difference is actually the protein that you think you measured. So it's an analytical validation, usually requires orthogonal methods of validation or measurement. And then there's also the clinical validation, which basically takes all your analytically validated biomarker candidates and it tests them against a very large cohort of clinical specimens. And so it's the clinical validation ultimately that serves to confirm or dispute your original hypothesis that one of these many differences is actually linked to a disease. It's actually quite interesting. Many of the biomarkers out there, they actually fail at the clinical validation step, just because maybe the clinical cohort being used was not proper, maybe because there was a bias in selecting patients. So all this can lead to failure of clinical validation. And so discovery experiments in a clinical diagnostic assay development definitely can generate many candidate biomarkers, which will ultimately need to be analytically and clinically validated. And actually, in the whole commercial discovery process, the validation phase is probably the most expensive phase of any biomarker development and diagnostic assay development project. And then lastly, once you have a clinically validated biomarker or panel of biomarkers, you then would go onto diagnostic assay development. And so for diagnostic assay development, what's really critical is you have to actually know what you're measuring. So you have to really test and make sure your analytical method is compatible with the specimen that is being generated in the clinic. And so for diagnostic assay development, accurate quantification with statistical confidence is really, really important. And so, some of the solutions are classical solutions, mostly based on ELISA assays, but more recently, there are next generation ELISA type platforms or mass spectrometry measurements that ultimately will hopefully result in more accurate clinical diagnostic assays. So just looking at the bottom part of the slide here, during discovery, ideally you want to measure everything that is in the sample. So you're using a broad mass spectrometry approach, whereas when you have a clinically relevant diagnostic assay, you generate generally many samples, but you have very few biomarkers that actually pass the validation test. So that summarizes the traditional biomarker research pipeline. So what I'd like to do next is focus on a biomarker discovery project that some of my clinical collaborators and myself performed many years ago. And so that showcases how you would go on to discovering a biomarker that is relevant to a particular disease. And so the disease of interest is chronic rhinosinusitis. And the common chronic is a very common chronic disease. It leads to impairment in quality of life, and also leads to significant healthcare costs. So this chronic rhinosinusitis is not the kind of sinusitis you face that is cured by antibiotics. It's actually a chronic condition that leads to patient continuously suffering from rhinosinusitis. And this particular disease, at the time, it wasn't really clear what actually causes that and how it distinguishes itself from a regular rhinosinusitis. And there's also, other than surgical procedures, there's no curative treatment, or there was no curative treatment at the time when the discovery was done. And it's actually the third most common chronic disease in ear and nose. And also, so what we then did was we decided to go to a small cohort of patients that consisted of healthy individuals, people suffering from rhinosinusitis, and people diagnosed as suffering from chronic rhinosinusitis. And so what we did was we employed a methodology that's called iTrack, which is a differential labeling technology that allows you to compare different samples using a mass spectrometer. And so the way you would perform an iTrack analysis is you would take a clinical sample. For example, the sample we used in our case was a mucus that was extracted after sinusoidal intubation. And this mucus was digested with an enzyme that generates smaller peptides. So the enzyme we used was trypsin, and the trypsin was used to generate smaller peptides that are compatible with mass spectrometry analysis. So once we had digested samples originating from different individuals into small tryptic peptides, we performed individual labeling, isobaric labeling of these samples with an iTrack label. So this particular label allows you to add a defined mass to each peptide, and this defined mass is able to actually define or allow you to assign which sample the peptide that have this additional mass attached to it, which sample these peptides originated from. And that is used, it's a pooling method, so you would basically then take your individually tagged peptides originating from different patients, and you would then combine them and perform separation using HPLC chromatography, and then you would use a mass spectrometer, an LC-MS, to determine what these peptides are. And then what allows you to do when you use an MS mode, which means you take these peptides and you break them down even further, it allows you actually to decide, firstly, that the mass spectrometer tells you which peptide it is, what the actual peptide sequence is, the amino acid sequence, but also it gives you the so-called iTrack isobaric mass tags, which tell you how abundant each peptide was in each sample. So for example, here, the identical peptide was most abundant in sample number 3, whereas in sample number 1, it was least abundant. So this methodology allows you to multiplex and simultaneously compare different samples. It's a common mass spectrometry approach. The reason multiplexing is useful because the actual analytical process of mass spectrometry is actually quite expensive. And so if you have to pay for operator time, multiplexing allows you to cheapen the process. And so this is actually what an HPLC trace looks like. So this graph basically shows the elution of pooled sample from a HPLC chromatography column. And so if we zoom in on the peptide that's eluted at 34 minutes, and we submit this peptide to mass spectrometer analysis, we actually can isolate the 3 strongest signals that correspond to a peptide that's characterized by this mass spectrogram, which actually defines the sequence of the peptide. And then if we zoom in on the reporter ions from the iTrack label, we now know exactly how abundant this particular peptide was in different individuals. So we see here individual 2 had the least abundance, whereas individual number 4 had the highest abundance in this particular test. And so this particular peptide actually corresponds to a prototypic peptide that is found in anti-leukoproteinase 1 precursor. And so by performing this analysis, the mass spectrometer allows you to tell how about, firstly, what types of peptides were found in the sample. It also allows you to determine which peptides were overexpressed or reduced in expression. And so, so that allows you to see, to study molecular differences between these 2 or more mucus samples. So if we actually look at the list of downregulated proteins, the protein that is downregulated, actually one of the most prominent proteins that's downregulated in chronic rhinosinusitis patients is actually the precursor to lysozyme C. And it's a protein which functions in the innate immune response. So it's actually a bacteriolytic enzyme that degrades certain microbes. And so having a reduction in this particular enzyme in the patient, it suggests or allows us to develop a hypothesis that maybe if this kind of lysozyme is reduced in a large number of patients, maybe these patients develop the condition because their innate immune response is negatively affected. The other protein which is downregulated in patients is the clear-cell phospholipid binding protein. And it's a potent inhibitor of phospholipase A2. And so here it's actually quite interesting because actually phospholipase A2 is a pro-inflammatory enzyme. And so, so if you inhibit this enzyme, you reduce inflammation. Whereas if you actually have a low concentration of the enzyme inhibitor, it may even promote inflammation. So that could be a reason for, a specific reason for that. So just by looking at molecular differences between healthy and diseased individuals, you're actually able to develop novel hypotheses that you could test during a future validation study. Similarly, we have seen upregulated proteins, for example, calgranulin B migration inhibitory factor related protein 14, which is an inhibitor of protein kinases. So maybe that actually also favors inflammation. Now, so basically, I just showed this type of effort to see, kind of, to showcase what is involved in discovering biomarkers. I think the most critical component is actually you want to discover something that is significant to disease. So the most important thing is you have to have samples derived from well-characterized patients, and you need very good analytical tools for screening. But then more important than the screening part is actually the analytical validation part, because it requires a different type of methods to go. And so current methods employed for the validation, actually showing the validation important to determine that these biomarkers are actually really the biomarkers you thought they were. And so the 2 methods that are currently employed are so-called SRM and MRM methods. It stands for single reaction monitoring or multiple reaction monitoring, which is a mass spectrometry method. And the other major validation tool is the ELISA assay or immunoassay, which is inexpensive, but that does require antibodies. I think the chief difference also is SRM and MRM are not very sensitive, whereas ELISAs can be exceptionally sensitive. So if your biomarker of interest is low abundance, which many of the oncology biomarkers are, you actually have to employ a method that is able to measure the potential biomarker of interest, for example, in early cancer detection. Quickly mentioning MRM type methodologies. So MRM is a mass spectrometry method. And in order to develop an MRM assay, you have to know the sequence of the protein of interest. So you then perform an in silico digest. So you're basically using your computer to generate all the possible tryptic peptides. And then you select the peptide of interest that is unique and that uniquely characterizes the protein of interest. And these type of peptides, they're called proteotypic peptides. And then you do a mass spectrometry detection and quantification. And in some instances, you may even do a mass spectrometry confirmation of the marker of interest. And so the benefit of this particular method is it does not require antibodies. However, it is not sensitive enough for some of the biomarkers. And so, so basically, multiple reaction monitoring can be very specific and sensitive for detecting components in a complex mixture. But in terms of analytical sensitivity, it may not go into the picogram per ml or sub-picogram per ml range that is required for some of the biomarkers that are of clinical significance. The MRM method has the largest linear dynamic range for quantification, and it's commonly performed by a mass spectrometrist as a follow-up, as an analytical validation. But it does require somewhat more sophisticated mass spectrometers that are relatively expensive. An example from my own previous work in the area of neurodegeneration and muscular degeneration, we developed a proteotypic assay that measures P97 peptides. P97 is a protein which is involved in muscle degeneration and neurodegeneration. And we developed an assay that allows us to measure a unique proteolytic product of P97 using a mass spectrometer. And this shows the complexity of the involvement. So we basically generate an HPLC trace. We show the actual mass spectrum. Then we show the extracted ion and the confirmatory MS-MS spectrum to confirm the sequence. And so you can employ these type of tests for analytically validating biomarkers. The other fundamentally different technology is actually immunoassay development. So if your research has generated a novel biomarker and you have antibodies that are available to recognize that biomarker, the first thing you would do is you would develop an immunoassay. So that could be an ELISA, or as shown in this particular example, it could be an AMP assay. And so what you would do is you would screen for antibodies that show good pairing. So for most immunoassays, you would want to choose a sandwich assay for added specificity and sensitivity. But there are many, many different antibodies available against your favorite potential biomarker of interest. And the best first strategy is to see which of these many antibodies in combination gives you the best signal-to-noise ratio. And so here, this particular example showcases the development of an anti-phospho-P38 assay. And you see that amongst many different antibodies that are commercially available, there's a smaller number here that shows a really good pairing. That means they give you a very good signal-to-noise ratio. These type of assays can be your traditional plate ELISA assay or the next generation type assay, such as the acoustic membrane microparticle assay. And this shows the principle of this type of assay technology, that you would take a homogeneous, which is based on homogeneous bead assays. So in this particular instance, you would take a magnetic bead that has capture antibodies that capture your analyte of interest, and you have a cognate detection antibody with a hapten tag attached to it. You would pre-mix or pre-incubate this type of analytical assay mix with your clinical sample of interest. And then you would take this mixture and flow it across a sensor. In this case, it's an acoustic sensor surface that measures the mass deposition, which can be is proportional to the amount of mass deposited to the membrane. And in this particular case, you would take the magnetic beads and reaction mix and use a magnet to attract all the beads to the sensor, to the acoustic sensor surface. And then you would disengage the magnet, and only specifically associated beads will remain at the sensor surface, whereas all non-specifically associated beads would flow away in a buffer stream. And the principle of an acoustic assay is based on the fact that when you have a resonant acoustic membrane, so the membrane resonates at a high frequency, and you deposit mass to this membrane, it will lead to a slowing of the vibration, which can be measured. And this measurement is directly proportional to the amount of protein deposited. So if you have a standard curve in these type of assays, it allows you, by comparing your analytical result, comparing it to your standard curve, it allows you to exactly determine the concentration of your analyte of interest in the sample. And these type of assays, they can be done in a sandwich format. So you basically have an antibody which is either directly or indirectly linked to a magnetic bead. These antibodies will recognize your biomarker or target protein of interest. And you have a cognate detection antibody, which has a hapten attached. And this hapten can form an interaction with an anti-hapten sensor surface. So that's the sub-principle of sandwich assays. You can use competition assays or immunocompetitive assays in cases where you only have a single antibody against your antigen of interest, where you're dealing with a small molecule or a single epitope target proteins or biomarker analytes. And so it's a different assay format that can be used to measure that. Now, very often this one example of an acoustic immunoassay against the IL-6 analyte, and that shows you assay parameters such as the lower limit of detection. So using these type of assays, you can measure sub-picogram per ml levels of interesting biomarkers such as the pro-inflammatory cytokine interleukin-6. And then important for assays are also validation criteria. For example, you want your assay to show a certain characteristic that indicate the assay is not easily interfered by other analytes that may be contaminating a sample. So you have, during the development of these type of assays, you use specific criteria to rule out your assay measures other proteins of interest. And just to showcase some of the discovery studies that have been done, that have appeared in the recent scientific literature, I wanted to highlight two specific studies here. The first study is by Adona et al., which actually describes a discovery effort to discover potentially novel biomarkers in a myocardial infarction. Now, traditionally, what has happened is most discovery efforts in heart disease, they focused on historic blood samples that were taken from healthy individuals and compared it to individuals that had a myocardial infarction event. However, it has always been very, very difficult to actually use blood or plasma to discover novel markers. Most importantly, that is, or the most likely explanation for that is that plasma and blood in general can be a very poor method of detecting biomarkers. And so this particular study, the reason I want to highlight it, actually shows something very interesting, and that is it's actually a study that was done in a procedure that induces heart attacks or controlled heart attacks in a certain type of patients. And so it's a procedure that exposes a small region in the patient's heart to ethanol to induce a myocardial infarction event, and then this particular study actually took blood from the patient immediately before the procedure, during the procedure, and after the procedure. And so you had a sample, like a healthy sample that described the patient's state before before planned myocardial infarction. And then it was able to generate several samples during and after the procedure. And so what you don't have to worry about in these so-called autologous type assay procedures is you actually don't have to worry about patient variability, which is actually quite significant. So meaning whenever you saw a difference in this particular patient in a plasma-based biomarker profile, you knew it was caused by the procedure. And so samples were taken basically baseline 10 minutes, 60 minutes, 240 minutes, and 1440 minutes. So that's a very, very good example, a powerful example, how you can use the same patient to describe differences that happen over time, like in this case, over the procedure. And but I don't really have much time to go into the study and list the reference later on that you could go and see how this particular discovery initiative was done. And so those kind of so-called autologous discovery efforts, they kind of neutralize the need for large patient cohorts, which you would have to do to be statistically significant if you didn't have the same patient or the same patient baseline. And that is because different people can have significant differences in biomarker profiles, especially in blood and plasma. The other study that I wanted to highlight is actually a biomarker discovery study that was performed by Whitaker and et al. And this one was a study that focused on discovering breast cancer biomarkers. And this particular study was quite different in the sense that it used a hypothesis-driven or rational approach to define what could potential biomarkers be that relate to breast cancer. And so this particular case, they did not do what many other people in the breast cancer field have done, that is comparing blood samples of healthy and diseased individuals. But in this particular case, the investigators asked the question, what has the literature shown? What are the proteins that may be relevant to progression of breast cancer? And so they basically came up with a candidate list that included proteins affiliated with breast cancer, that also included surface proteins commonly found on breast tumors. And then they developed mass spectrometer assays that selectively looked for these particular markers, these candidate markers, to see how many of those could actually be detected, and more importantly, how many of those would be differentially expressed and could discriminate between healthy and diseased individuals. And this is actually a study that wasn't done on patients, but on mouse models of breast cancer. And then after they came up with a candidate list, or they narrowed down the candidate list from almost 2,000 candidate markers to about 572 markers, they used SRM assays and immuno-SRM assays to actually come up with a kind of small list of biomarkers that are currently being further analyzed. So that's another way biomarker discovery these days has changed from only several years ago, in a sense that people use many more hypothesis-driven approaches as opposed to simple screening approaches. And just to show what it would take to perform something like this, you're actually looking at your initial discovery takes several months to perform. Targeted quantification takes another 5 months. And then ultimately coming up with good quantitative assays take anything close to a year, and the cost of these type of assays is actually quite significant. So the key for discovery and validation is that's something a lot of people underestimate, is you have to start with a well-defined patient and control cohorts, because actually there are many studies that have been performed over the last 10 years that showed that really simple procedures, such as, for example, taking blood from a patient where the patient is sitting up versus the patient reclined on a bed, just the position of drawing your blood can lead to significantly changed profiles. Concurrent with the sampling method, you need unified strict sampling procedures. For example, when you perform a surgical extraction of a tumor, it makes a huge difference if the tumor is immediately snap frozen, or if it sits on in an operating room for 20 minutes or half hour. So not having these type of processes like sampling defined is actually a recipe for a poor differential discovery of biomarkers. So it's really, really critical, strict sample preparation methods. Also, if a patient allows for the possibility of autogenic specimen collection, that actually can enhance the discovery initiative, because you're eliminating genomic and phenotypic differences. And then one thing to always remember is in proteomic discovery, patient individuality, sampling and sample preparation can account for the majority of proteomic differences. And so ultimately, all these potential differences have to be validated to be really certain or to ascertain that these differences are actually due to the disease. And very often, even for blood-based biomarkers, people now go to use tissue that are relevant to the disease, assuming that some of the tissue proteins are shed either directly or through exosomes. And that avoids the blind screening of plasma, which historically has been very, very difficult. So last few minutes, I wanted to touch on aspects of clinical validation. Clinical validation is defined as actually screening the number of patients that have been diagnosed with a relevant disease, such as cancer or specific subtype of cancer. And basically, in order to do clinical validation, you have to build assays that allow you to do so. And these analytical assays, many of these assays actually require the selection and characterization of antibodies that faithfully can measure your candidate biomarker in a clinical validation study. Generally, it requires large screens to select antibody species that can recognize the analyte and that are not affected by other matrix interferences that might be present in your sample. It can require extensive kinetic characterization of antibody affinity, avidity, selectivity, and specificity. And some instances, you may actually have to synthesize or make an antibody that needs to be extensively characterized, because sometimes conjugation can affect antibody properties. And so it just shows that actually a clinical validation, although it's very intuitive, actually the fundamental work required to build assays is actually quite immense, and that links to the cost of developed assays. And most important to remember is that most of the biomarker candidates, they actually fail at the stage of clinical validation. And so that imposes the question, if you and your particular team or effort has one or two biomarkers that may relate to a clinical case, or may relate to a specific type of cancer, developing assays for those biomarkers, there's no guarantee that actually these biomarkers will really be linked to the disease. And so larger initiatives, maybe community-driven initiatives that take a whole collection of candidate biomarkers from different studies, and developing a validation cohort could really streamline the process. So you don't have to individually use a well-defined patient and use up rare disease material, but you could maximize the impact by taking the whole community biomarker candidates and then looking at one really, really well-characterized cohort to see if one or more or any of these candidate markers hold the test of clinical validation. Once your biomarker has passed the test of clinical validation, you would generally develop a diagnostic test. And so that can be a test that is either run in a service lab, such as a CLIA laboratory, or it can be a test that can be offered on a approved diagnostic platform. And so most of the current proteomic diagnostic tests related to cancer, it actually requires a selection characterization of antibodies, because most of these tests are antibody-based. They require a very, very specific assay characterization, so to understand interferences, because you want to make sure when you have a clinical assay that your assay actually measures the analyte of interest. And so there are established methodologies that allow you to do so, to basically define the degree of interference and the profile of interference that may affect your immunoassay of clinical significance. It requires the following of established validation guidelines. And many of these assays can be run under a CAP-certified CLIA lab. So that's something many companies do with in the oncology field. They like to run their assays under CLIA first, because it actually gives them the opportunity to do a lot of development work, but also to get more experience with the type of assay that has been run, and that can lead to better validation of the assay. And then once the assay has been properly validated against a larger cohort of individuals, these assays could be offered as a 510K type approved assays, but very often coupled to a diagnostic device. So just in summary, the promise of predictive and diagnostic biomarkers is manifold. They're better diagnosis and early intervention is really important when you treat cancer, because very often cancer is an individual disease that is made up of individual genomic and proteomic differences, and only recognizing and measuring these differences will have a major impact on patient treatment. Also, biomarkers are required for more efficient drug development. So, for example, patient stratification. And lastly, you can, by having biomarker-driven type diagnostic and therapeutic approaches, they could really significantly eliminate the empirical approach that I mentioned earlier on. So just wanted to give you a number of key papers for further reading. If you'd like to go more into the biomarker discovery type approaches, I think in terms of discovery of biomarkers, I'd like to highlight the two papers by Adona et al. That were published in Nature Biotechnology in 2011, and by Whitaker et al. Also published in the same issue of Nature Biotech. And then also for clinical assay development, and unfortunately didn't really have much time to delve into that today, I wanted to highlight the paper by Taguchi and Hanash that really very nicely outline the steps required for actual clinical diagnostic assay development in the area of cancer. I'd like to acknowledge all past and present members of my research team, some of which contributed to the slides that are shown today on discovery and immunoassay development. And I'd like to leave you with the continuing education URL. And so that is something, if you want to obtain your continuing education credits, you would go to this particular link. I'd like to stop here and take any questions. The first question is actually, what is the minimum sample size required to validate a biomarker as a diagnostic tool? It's a very, very interesting question, and the answer is, well, the simple answer is as many as possible, because you only will see, you only have really good statistical power the more samples you have. In many instances in the world of oncology, that is not very practical. And so generally what I've seen in the discovery initiatives is you would start discovery of biomarkers in a relatively small cohort of patients. So if it's a common cancer, I typically see somewhere between 40 and 50 individuals that have the disease, and maybe between 20 and 40 healthy equivalents. And those usually make up discovery cohorts. Now, it also depends on the genetic or proteomic diversity of the cancer patient versus the healthy individuals. So, for example, in breast cancer, where there are many, many different molecularly defined cancers, the discovery cohorts would have to be significantly larger. And so I think, the answer is, if it's a relatively homogeneous type of tumor, you're looking in different individuals, you look in the dozens, but ultimately you'll have to look in larger cohorts. Now, there are number of instances, for example, in pediatric cancers, the cancer is relatively rare, and so it becomes very, very difficult to actually do these large discovery initiatives. And so very often what you would want to do is you take some really well-characterized patient, and then you see, can you obtain autologous material? For example, can you extract part of the tumor and extract part of an equivalent tissue that mimics the developmental stage and program of the actual tumor. In best instances, of course, you always look at autologous type samples, in which case you actually can use a lower number of cohorts. The other question is, can you think of successful examples of transcript expression as biomarkers or as protein level, a more accurate measure of biological events such as disease development and progression? A very interesting question as well, because cancer is actually defined as a genetic disease. And so it's actually very straightforward to look for translocations that are associated with tumors or mutations that in signal transduction components that relate to a progression of cancer. And so these kind of genomic examples definitely exist. They're actually part of a diagnostic or clinical practice today. For example, one assay that uses transcripts is the Genomic Health marketed assay, which looks for transcripts that allow you to discriminate between different types of breast cancer. Now, the field of breast cancer is a good example where you basically need both a genomic and proteomic components. For example, you would use a genomic health profile looking at transcripts, whereas you would use a immunohistochemistry type methodology to actually investigate the HER2 status of a tumor. So I think in oncology, both methods are equally well used. I think there's from a genomic point of view, it's very often easier to look at genomic differences. However, there's certain proteomic differences that are linked to the disease and the etiology of the disease. For example, HER2, classical example in oncology. And also when you make a treatment decision, very often it becomes important to actually understand which of the many possible signal transduction pathways are responsible for growth addiction of the tumor. And so by understanding the proteomic profile or the signal transduction nodes of these type of tumors, it should make making treatment decisions easier, as opposed to just looking at a transcript. So I would say, in summary, it very much depends on the tumor and the type of decision that is being made. In a number of cases, there's a developing trend that goes towards looking into the sort of classical plasma-based proteomic biomarkers that indicate the presence and absence of a tumor. For example, there's effort in individuals that have ovarian cancer to actually establish a baseline of CA125, which is a classical marker for ovarian cancer, and you would follow the diminishment of CA125 level as the tumor responds to treatment. It's very difficult to use CA125 as a pure diagnostic marker, because individuals actually can vary with their basal CA125 level. But actually, if you follow individuals over time, you could make a certain kind of a decision based on these type of proteomic markers. Then no more questions. I'd like to thank the audience and the organizers, and I'd be happy to take additional questions by email.